will stop sharing when I start sharing.
Good morning, everyone, and welcome to day 2 of our Cocoa Research Center Annual Research and Development Symposium 2023. Welcome back to those who were here with us yesterday, and welcome to our new attendees. Today, we have a number of exciting presentation plans for you guys, and we look forward to executing them and building a better understanding of what is involved at our Cocoa Research Center. All right, so first up, I would like to welcome Professor Haran to give us some introductory announcements as well as our welcome. So Prof. Haran, can you please share your presentation and introduce?
I think the host has disabled sharing, so I need to enable the sharing, please. Right. Good day, everyone, and welcome back to day 2 again. My initial introductory talk today is really on the topic of building an impactful, sustainable cacao value chain. And this I'm going to look at in terms of the functions of a research institution such as the Cocoa Research Center, what it can do in support of building such a sustainable cacao value chain. Now to start with, I would like to start with some of the global challenges that we face. And as a result, as a group, we need to try to understand these problems and be able to solve them, right? So starting with climate change, we don't need any introduction. It's something that we are facing every day. And in Trinidad and Tobago, the last couple of years was really bad. We had a very low production. Diseases, and most countries that are cocoa growing would know the impact of diseases can take out about 40% to as much as 80% losses. And that can make or break the cocoa farming business model. We talked about also cadmium in some of the past presentations. It's an emerging problem in Latin America and the Caribbean region. Without proper mitigation methods, you cannot import the cocoa. So cadmium is one of many food safety problems that we would be facing. And then the global crisis, the food crisis that we are facing. So these are global challenges that we need to really look at in a holistic way so that as research institutions, private sector, NGOs, we can tackle this problem in a sort of coordinated manner. The chocolate market, the Trinidad and Tobago and the Caribbean region and parts of Latin America and the rest of the world, we market in the niche high value market segment. And this segment itself is evolving. And as it evolves, as a country, we need to reconfigure our cocoa value chain to meet some of those new requirements or exacting requirements of the industry. And then you would have heard about the sustainable development goals that we are all trying to achieve. And then, so my talk is really going to see how, as research institutions, we can address these questions. And I have 5 talking points today, 15 minutes. So I'm going to first tell you a little bit about the Cocoa Research Center and what we are doing, how we are structured. I'm going to talk about the planting material pipeline that is required to sort of innovate in the cocoa value chain, talking about some of the mainstream of R&D innovations that has to happen in production, processing, and marketing. And then what kind of innovative investment environment that we need to foster to create these new novel challenges, and how do you really build a marketplace that values some of the work that comes out of these research and developmental institutions. So to start with the Cocoa Research Center, we have a research center established 93 years ago exact. And we have been doing research along the whole value chain, but more at the level of planting material end because of the International Cocoa Gene Bank that we curate. This is the gene bank. And then recently we set up an innovation center so that some of the research that we do at the Cocoa Research Center can be converted into innovations, technologies, and they could be commercialized into the commercial environment. So that is what we are trying to do. And that is our sort of framework model that we are using to make that reconfiguration of our research center. The first topic that I said we need to talk about is partnerships. We cannot do it on our own. We need to have various kinds of partnerships. And one of the first partnerships that is important is research partnerships. Very often research centers themselves are not completely adequate. There are gaps, weaknesses in each research center. And only through collaboration between research centers can we achieve the larger goals, particularly that face the industry globally. And so we have been trying to build such linkages between universities across the globe, mainly in Europe and North America, I would say, but also with Latin American countries as well. The second type of partnership is very important, is the industry-university partnerships. Now, this is very important because the industry in all its forms, starting from the private sector companies, the NGOs that operate in the environment, they are the ones who are on the ground and experience some of these challenges. And having partnerships between research and industry is extremely valuable because we could make or do research and create innovations that actually can make a difference on the ground. So that is very important. And the third, I would say, is government research partnerships. When I say government, it could be multilateral public funding coming from global organizations, or it could be the government of the country that you're working in, and this kind of partnership is very important, particularly to solve local or sometimes international problems, that face the countries or the sector as a whole. And then finally, I come to the triple helix model of innovations. And this is really particularly interesting in disruptive innovations that we do. If you have doing research and we come across new technologies, these technologies have to be commercialized, and very often these could be disruptive. And you need, private sector itself is not usually in a position to take risks. And this is where I think, the public-private university partnerships become extremely important. So these are some, in the last year's presentation, I talked a lot about the planting material pipeline. This is the innovations that we need at the gene bank end to provide material that meets the need of farmers across the world, and what sort of innovations can happen at the gene bank end, and what innovations need to happen at the country level, to be able to have this pipeline flowing smoothly, producing new materials all the time to support farms. So I'm not going to spend too much time because we talked about that in the last one. The second is creating that innovation infrastructure to solve some of these larger problems in production problems, marketing problems that we face. And increasingly, that people are very much worried about the carbon emissions, and they are all looking towards building a carbon neutral industry. And cocoa industry in itself is not very good at doing that. I think for every kilogram of chocolates we produce, it's between 5 to 7 kilograms of carbon dioxide is released into the environment. So there has to be a lot of changes or reconfiguration that has to happen from the farm end, the production end, and the whole value chain up to marketing. And that needs a lot of technological innovations. And we have constructed the International Fine Cocoa Innovation Center as our innovation apparatus that sort of links some of the global problems and creating the innovations needed to take them to the next level. But I think as all research institutions should have that sort of mechanism in place so that we can solve some real life problems that industry is facing. And then when you want to make these sort of reconfigurations of the industry, you need investment. And that is very important. In all countries that grow cocoa, there needs to be reconfiguration because the market is changing, the demands of the market are changing, the global challenges are only in the increase. And we need to do reconfigurations, and that needs investment. And to attract investment, you need to do several things. And I've just listed some of the things that I think are very important. These are some of the issues we are grappling with in Trinidad and Tobago in terms of attracting investment, creating the right policy environment, creating attractive business models out of these cocoa and chocolate and other value-added outfits so that they are attractive to businesses. Creating and building carbon neutral supply chains, mitigating risks so that investment will flow in, because when you have your risk is very high, biological or environmental, the investments would be very hard to come by. But if you can minimize those risks, then it could create. And then you have these general things like you need to have proper technical assistance programs, you need to have better farm management expertise, you need to have private sector services so that the industry can grow and flourish, right? So these are some of the things that we need to do to attract that investment required to reconfigure the industry. And this is some of the things that we have been doing. And yesterday you would have heard of a presentation from Marvin, who talked about mapping of these risks. So we have been going around the country and mapping the various risks so that we can provide risk mitigation measures where risks are high, and as a result, make those environments more lucrative for investments to happen. And then finally, the partnership I talked about was the triple helix model, right? The triple helix model, I already said, is that new innovative innovations, commercialization sometimes is risky. I think the private sector considers them risky because they are new, they have not been tested, there are no markets for those new innovative methods as yet. And very often, they are just at the proof of concept stage. And very often you need pilot scale innovations and then scaling up innovations. And this is where the public sector funding is very much important, because these innovations will stay at the incubation stage and never find a market if the public sector does not invest, de-risk some of those investments so that these new innovations can actually find a successful implementation on them. So that said, I would like to stop here. And this is And most of the presentations that we are going to be doing today will be tackling some of the work that we have been doing in this environment to create innovations, to solve some of the problems that we face locally and internationally. So with that, I will say thank you, and I hope you enjoy the rest of the presentations.
All right, thank you very much, Professor Yuma Haran, the Director of the Cocoa Research Center, for that enlightening introduction on the overview of the ongoing activities at our center. For everyone who may have missed it, I will be your moderator today. My name is Dr. Gideon Ramtahal, and as Professor Yuma Haran would have mentioned previously, we have a number of exciting presentations planned for you all today. We will be picking up from where we left off yesterday on the topic of fine flavor quality management, with 2 presentations planned for this particular area, followed by 4 presentations on the topic of genetic resources and conservation. If you also missed it, we have some brief presentation guidelines for presenters. Please note that you have 15 minutes for each of your presentations, followed by 5 minutes of questions from the audience. I'm not too sure if anyone has any questions for Professor Yuma Haran before we move on.
Yeah, I think there's one question from Mark Hume. It says, thanks for the nice introduction. Are you looking at other aspects of sustainability, such as deforestation, biodiversity conservation, and retention of other ecosystem services? Yes, of course. I think we are implementing a project exactly on that, trying to understand some of those factors that provide ecosystem services. Cocoa, as you know, is not only producing cocoa, but it's grown in very vulnerable environments, very often slopes, watershed management. They provide a lot of environmental services, carbon capture, watershed management, soil conservation, and so on. And we are looking at all of those, including biodiversity conservation, as a means of developing certification and traceability of these farms that have good ecosystem services, so that they can be marketed to the final producer. So thank you for that question.
Right. Are there any more questions? I'm not seeing any more questions in the question and answer tab, and no others in the chat. So Professor Yuma Haran, thank you so much for your introduction. And we will now proceed with the other presentations for this morning. Okay, ladies and gentlemen, first up, we have Dr. Lambert Motilal. He is the lead of genetics and genomics team working on gene bank fidelity and conservation genetics. And today he will be showcasing the use of genetics in the fine flavor industry, which is a presentation on genetic diversity helps provenance of premium cacao. Dr. Lambert, the floor is yours.
Good morning. This group of 36 international collaborators will now make a case for genetic provenance in the cocoa industry. The ICCO has the 2 definitions of fine cocoa and flavor cocoa, and both of them relies on this phrase, may also offer important genetic diversity. But what is meant by important genetic diversity? And how do we sample and describe genetic diversity? Well, in cocoa, at present, we can fit the ancestry, the genetic background of all cocoa accessions into 10 groups. And of these 10 groups, there are 2 groups, the Criollo group and the national group, that are alleged or are known to have fine flavor and command premium prices. So then we can ask ourselves, how are these groups represented in countries that have premium cocoa? So instead of saying fine cocoa or flavor cocoa or final flavor cocoa, let's say premium cocoa, cocoa that's guaranteed to bring you premium prices. Just a minute. We can describe genetic diversity in several ways. We can use a cluster diagram or a tree diagram. We can look at the actual ancestry estimates within each of the 10 groups or any known accession. And of course, we can have a match analysis to compare varieties among themselves and to known reference varieties. And this fingerprinting service is available at the Cocoa Research Center at a cost of $51 per sample. So if we look at the different genetic diversity, we look at premium cocoa, but what is this provenance? We make a difference between provenience and provenance, where provenance is just so much more. And we ask ourselves, how can we use genetic diversity to help provenance in premium cocoa? And how does the genetic diversity among fine flavor countries differ? So let us look at Ecuador as an example. On the web, you may or may not have seen the chocolate journalist, and the chocolate journalist writes that the national cocoa, which is a premium cocoa, is rapidly disappearing. And also it is considered a Criollo strain. And that we should pay, let's say, for instance, a premium price for national cocoa. So we agree that yes, national cocoa is a premium cocoa, but genetic diversity says it is not Criollo. In fact, it's two different types, right? So we can use our SNP fingerprinting panel to authenticate national trees. And now we answer the question, if national cocoa is disappearing, do we have enough national genetic resources conserved? So in Trinidad, at the Mapa farm, in the early years of cocoa collection from the Amazon, germplasm was collected and brought back and established at Mapa farm, including national types. This farm was recently revisited from a team at CRC. We went in within this sort of secondary jungle and resampled and retyped these trees using our SNP fingerprinting panel. And we found at least 200 trees had at least 75% national ancestry. So enough national trees, enough national cultivars are conserved in our gene bank collection. And of course, in Ecuador, where national originated, they would have additional national trees conserved. So what it tells us is that if national is a good flavor cocoa and high flavor cocoa, as is known, then let us use national trees in our breeding program in Trinidad to improve the complexity of Trinidad flavor profile. If we were to release trees to farmers, we first must know their field performance in order to give farmers correct and well-yielding genotypes. So let us test national trees for their field performance. Okay, so what about these fine flavor countries? What about the farms in these countries that command premium prices? What do they have? Do they have accessions with national and Criollo in it? And if so, how much? So we looked at 11 countries that took advantage of our cocoa fingerprinting practice, right? With various number of samples depending on the client's preference. And of these 11 countries, 5 are actually on the ICCO list as final flavor producing countries. The first thing we did was to look to see a match analysis. That is to see if we were to check these samples that originated from these farms in particular countries, do they match up to any known variety? And in fact, some did, of various types from different countries. The first 5 you see are like TSH types, so let's say TSH 919, TSH 1188, ICS type like ICS1, ICS60, right? And the first 5 can be considered as fine flavor varieties, but the other 5 currently are not considered as fine flavor varieties. So it tells us that within these countries and within these farms that provide premium cocoa, that not always do we get fine flavor accessions. Let us look now at the genetic diversity of these countries as compared to each other and the 10 groups of cocoa. And we notice that they all occupy different spaces within this PCOA, Principal Coordinate Analysis graph, right? So they occupy different spaces, so they have different genetic diversities. Here we have Trinidad close to the middle, and all the countries we've done is triangulated amongst Amelinado, Criollo, and Nacional. Let us look now at the phylogenetic tree where country as a unit is represented, and we can easily see that Ecuador has a lot of national trees. Philippines have a lot of Criollo type trees. Dominica has a lot of Amelinado type trees. And you notice again that each country occupies a different space. Some are more closely related to each other depending on the genetic mix of the accessions that they have or that they have sampled as elite trees. And what now of the individual samples? So we're going to look at a few phylogenetic trees. There'll be 2 trees per slide. And on each tree, the 10 genetic groups are represented in different colors. And always Criollo will be at the top in blue. Amelinado will be in dark green, and Nacional will be in orange. In addition, we have 2 reference cultivars, ICS1 and CCN51, in each phylogenetic tree. And each tree will have a different combination of countries for ease of presentation. And the countries will be color-coded as you would see here, okay? So this tree deals with the Philippines, Hawaii, Venezuela, and St. Vincent. And what you will notice immediately is that each country occupies or has samples throughout the entire phylogenetic tree. Some are between Amelinado and Criollo, some are between clusters, there are few within clusters, right? And each country has a different sort of distribution profile. If we look at Peru and Trinidad, which many more samples to consider, you will see the distribution is different. Peru has samples that border Nacional group and also from the Amelinado group. And Trinidad have samples with Amelinado as well, but a lot more between Amelinado and Criollo. If we look at Dominica and Jamaica, we see that Jamaica has, apart from the fact there are much more samples that were done from Jamaica between the Criollo group and the Amelinado group. But Dominica has more with around the Amelinado group and few closer to the Criollo group. So if we look now to try to estimate the actual ancestry level and not just the relative positioning, we would find that very few pure accession, that is a cultivar that has membership in only one genetic group, is present among the countries. Some countries have no pure accession sample in the elite trees like St. Vincent and Venezuela, Bolivia and Hawaii, right? El Salvador and Philippines had one Criollo accession. That is to say there's one accession represented there as 100% Criollo, but there may be different trees or different samples having the same match identity. So it's one type, but you could have 10, 20, 30, 40, how many trees according to its match analysis. Within Ecuador, we see we have 11 national trees, high quality, high percentage national trees, and these were all 100% national. In Dominica, we had 10 trees that were 100% Amelinado. In Jamaica, 10 trees, 100% within this group Marignan. So even in the 5 flavor countries, in these countries with premium cocoa, what you have accessions of varieties that are represented on farm that has a single ancestral origin, but may not be and definitely are not Criollo or Nacional in some of the countries. If we to look now at the hybrids, because if we don't have pure samples we have one pure sample per country, and that country sent in 500 samples, then 499 of these are mixed individuals. So the majority of these elite premium cocoa trees are mixed individuals. And how are they mixed? Well, they're mixed mainly with Amelinado and Criollo, or Amelinado Criollo and something else, or Amelinado Criollo Nacional and something else. And in Ecuador, at present, it's the only country that has the combination Amelinado and Nacional. But aside from having these particular combinations, there are also combinations that do not have at all or very, very low percentage ancestry of Amelinado, Criollo, and Nacional. So for example, we could have Contamina and any other combination, very low ancestry of Amelinado and any other combination, Iquitos and any other combination, and so on. So in general, we can make 4 main findings. There's a wide genetic diversity of cocoa within and among countries with premium cocoa. Fine flavor can come from many different genotypes. Even 100% fine flavor countries have cocoa without Criollo and Nacional ancestry. So that means fine flavor, premium flavor is coming from groups, ancestral groups other than Criollo and Nacional. And in fact, this has been supported by results within CRC from Dr. Naila Ali, who has looked at flavor differences using only pure accessions in particular groups and doing just pure group fermentation. So we know that fine flavor can come from group other than Criollo and Nacional. So maybe we should think of calling cocoa that commands high prices premium cocoa, of which you could have particular different classes, whether it's origin cocoa or organic cocoa. They all become premium cocoa, commanding a premium price. So what do we recommend? We can have 4 main recommendations. We say, let us investigate whether there's a minimum level of Criollo and National ancestry that can guarantee fine flavor. This becomes part of your provenance package. So then you can say, yes, on farm, I have this amount of trees with Criollo ancestry, and they all have Criollo ancestry, and the Criollo ancestry is at least 40%. That's a good selling point. Let us investigate the flavor and genetic profile of individual TSH cultivars. And this is important for Trinidad because we have this TSH breeding program that has generated a lot of TSH clones, many of which have gone throughout the world, right? But let us see exactly what is the genetic profile of each individual TSH cultivar, and let us see the flavor profile of each particular TSH cultivar to know how best to use them on farm. And these national cultivars, well, let us have a nice deployment of these cultivars, but first we must know their field performance. So let us assess them in field to see what, if any, can they be used towards being used on farm as farm material to bring in additional revenue for farmers as having premium grade cocoa. We should use the cocoa DNA fingerprinting service at CRC to describe on-farm cocoa genetic diversity as part of a provenance package for origin cocoa and specialty cocoa. We don't need to do it for bulk cocoa, but if you want to show that your cocoa on farm should have a premium price, this is one of the supporting documentation that they can use. So therefore, we should be able to use genetic diversity correctly to help decide farm gate price for specialty cocoa. And lastly, government should fingerprint all their elite cocoa varieties, as well as conduct a genetic survey of their cocoa countrywide. So in closing, I would like to thank the institutions, farms, and individuals who help in this work. Thanks to our donors, and thanks to you all for considering genetic provenance in cocoa. I thank you.
Thank you very much, Dr. Motilal, for your presentation demonstrating how genetics can be used to support continuance of fine or flavor cocoa within our region. Now we're looking for questions. I recall seeing a comment in the chat from Mr. Nick Davis. He says, but if the samples are taken from select farms where breeding programs were in place historically, doesn't that throw out all the results? Lambert?
Okay, thank you, Nick. The situation is that not in all countries, and definitely not in all farms, would there deliberate cocoa breeding programs, right? In some places, farmers would select their own seed material for plant and to grow out. And not in every case with the repeated waves of introduction, not just for germplasm transfer, but also from ministry-led or say government-led introductions, there have been a change in the farm composition, right? And not always have we really paid attention to this genetic mix on farm and how it contributes to the fermentation profile or the flavor profile. What we need to have is a clear understanding of all the varieties and their ancestral background, all their genetic mixes. What are we really working with? So I would say it does not throw out all the samples, but it's something we need to be careful of when we select them. In this study, we dealt with elite trees. These are the trees that the actual farms, the farmers, the groups, the industries prefer to keep on to use as planting material. And therefore, these are the trees that we look to command in high premium prices.
All right, thank you. We have another question from Samuel in Haiti. Did you ever consider including Haiti in those research?
That's a very interesting question. We've worked with some projects in Haiti before via the Catholic Relief Service and the Lutheran World Relief Organization, right? And we are actually processing some samples right now. The issue we have and that we're very cognizant of is confidentiality. And we've only presented a bit of the story here from select persons who were asked whether or not they would like to showcase their results. We did not do the samples from Haiti because I did not ask them that, and because the number of samples there are yet to be processed.
Right, we have another question from Ian, and they asked, very interesting investigation, the cocoa flavor in different countries. Did you collect some samples from Indonesia?
Is that from Chengxing?
Yes.
Yeah. Hi, Chengxing. Good morning. I would love to go to Indonesia to collect samples, you know. What we have is that the clients provide the samples to us. So if there are clients in Indonesia willing to provide samples, the cost, remember, is $51 per sample. We give you a nice report with everything we've done here, from the match analysis to ancestry analysis. We would really love to have samples collected from all different countries throughout the world, including India and Sri Lanka, places where you have relic material, especially relic Criollo material. We did a few, we are going to do a few from the Philippines. We'd like to get some more. So any country, any farm, any industry with access to cocoa trees that you'd like to have fingerprinted to know their ancestral background, please do send across.
Okay, we have another question from Daniel Paruma and from Bolivia, sending his greetings. He said his query is whether these genetic groups that our country shares may be potential for denomination of origin, considering that they are mostly hybrids.
Hi, Daniel. Hello. I'm not too sure what you mean by hybrids for these, when you say these groups. Remember, the convention right now is that we have 10 genetic clusters. That will change as we encounter more samples, especially from the wild, from the Amazon, right? So we might have more than 10 clusters. It means that no matter which cocoa tree we were to sample to assess, that cocoa tree can have a background from any one of these clusters, right? It could have only one cluster or a combination from any of the number of clusters that exist. So the groups themselves are not mixed. The cocoa varieties, the accession, the farm accessions can be mixed, right? And what is important to know is how they are mixed. because how they are mixed can let you know how you want to ferment. Are you going to ferment everything en masse, or are you going to do particular fermentation, which becomes more origin or niche fermentation? So are you going to ferment by a plot, by a field, by an estate? Are you going to ferment by a particular type genetic origin? These are the things you can do now, knowing the genetic profile that we can provide to you.
Okay, and we can take about one more question before we move on to the other presentation. We have Dave Lloyd asking, how would you advise that this data is used to guide cocoa propagation, especially in terms of selecting rootstock and scion material and the pollination type of these cultivars?
Hi, Dr. Davison. Pleased to hear from you. Yes, well, definitely what we have to always do is be twinned, right? Or more than twinned, yeah? Because the research only offers one part of the picture. There is just so much more we need to be able to do with it. So when we identify promising trees, these promising trees right now is only based on genetics, right? It's only based on the profile. What is needed is the accessory information. Are these productive trees? Then those becomes the ones to conserve and propagate and provide to farmers. Knowing the ones that you find interesting, those are the ones now to go in the field, go on farm and check to see what favorable traits do they have, and therefore what can I leverage from that, and therefore decide how to use it. So we say this is a tree, and this tree has, let's say, 40% Criollo. We say, okay, chances of having fine flavor is good, right? But how does it compare for disease status? Is it prone to diseases? You may want to be careful in how you actually go about implementing your choices. So accessory information is needed to decide for these other ventures.
Right. Thank you very much, Dr. Motilal. Just a reminder, guys, that if we are unable to capture all of your questions within the time frame, we ask the presenters to facilitate the responses online.
Sure.
Appreciated. All right. So up next, we have another presentation under the fine flavor quality management subject area. And we have Dr. David Gopalchan. Now, Dr. David Gopalchan is a research fellow at the University of Nottingham. He has extensive expertise in molecular biology, genomics, and bioinformatics, as well as understanding microbial communities, which he'll be speaking about today. Before joining Nottingham, he served as a research fellow at the Cocoa Research Center, and he will be exploring genetic diversity within cocoa populations. I would like you all to welcome Dr. Gopalchan, who will be presenting on the topic entitled Chocolate Symphony, the Hidden World of Microbial Interactions in Cocoa Fermentation. Dr. Gopalchan.
Hi, good day everyone, and thanks for joining in. So in this talk, I'll be telling you about some of the work we've been doing around cocoa fermentation. So during this conference, several of the speakers spoke about the quality and flavor of beans. So for instance, Lambert told you about the cocoa varieties and essentially the genetic effect on the quality and flavor, while Darren and Nyla spoke about human interactions and how it can affect the quality and flavor of the beans. In this talk, I'm going to focus mainly on the microbiome. Now, the microbiome is a major component in the environment. And in fact, it's likely the underlying mechanism behind many of the environmental factors that affect the quality and flavor of the beans. So a number of factors affect the flavor of beans. However, the fermentation process is a critical step. So it's a natural process driven by microbial communities that reduce the bitterness of the beans and help develop the flavor precursors. Now, fermentations are typically performed on the cocoa farm after pods have been harvested. And currently, we depend on the natural microbiota from the farm to ferment our beans. So cocoa beans from Colombia are well known for their desirable flavor. flavors, with beans from Huila, Santander, and Antioquia. These are 3 cocoa growing regions in Colombia, are known to have very distinct flavor profile from the other. So the question is why? Why do we get these differences in flavors? And could it be related to the fermentations and the microbes in the fermentations? So to answer this, we went to a farm in the Santander region, and we analyzed the fermentations over 2 harvest periods. We looked at the temperature, pH, and color changes of the beans. So initially, there were very little changes in the temperature of the beans, but this was followed by a rapid increase after about 2 days, which then plateaued to about 50 degrees. There were corresponding changes in the pH. So the pH of the pulp initially was a little above 3.5, but then as the fermentation progressed, we saw a linear increase in the pH. In contrast, the pH of the cotyledon was a little above 6. After about 2 to 3 days, then we saw a drastic decline in the pH. We did see a strong correlation between the temperature and the pulp pH, right? And this was a strong positive correlation. Analyzing the distribution of these parameters, we found that the temperature and the pulp pH were linked, while on the other hand, temperature and cotyledon pH, while the cotyledon pH and pulp pH were not linked, indicating that the fermentation was a bit compartmentalized, and the fermentation in the bean, the cotyledon versus the pulp were happening a bit differently. And these changes were accompanied by changes in the color of the beans, from cream to sort of brown red towards the end of the fermentation. To analyze the microbes, we did Oxford nanopore shotgun DNA sequencing. So we saw that the microbial communities were most diverse at the start of the fermentation, but then the diversity of the communities decreased as the fermentation progressed. And we saw this with both bacteria and fungi. The microbial communities were also quite different at different days of the fermentation. And the phylograms here shows the most abundant families that we identified in the fermentations. We identified groups of microbes that changed during the fermentation. So this is with both bacteria and fungi. So we saw microbes that increased, so became enriched as the fermentation progressed, and those that became depleted with the fermentation. Now, to understand the fermentation in a broader context, and to understand the link between the flavor development and the microbes, we then analyzed fermentations at the other 2 regions, in Huila and Antioquia. We first, however, when we went to these other locations, we first verified that the varieties that grew on their farms were similar to the varieties to the first farm we visited in Santander. So we did SNP DNA fingerprinting. So this PCOA of genetic distances shows the Colombian sample. So this is from the 3 farms fell within this region here. So it's close between the Melanado and Criollo group. The ancestry analysis confirmed this, showed that the varieties grown on the 3 farms were mainly a Melanado Criollo hybrids. Although we detected other backgrounds present, for instance, Contamina was also there. There was also some Iquitos, Marinon was also present in these varieties. But overall, though, the genetic backgrounds for the varieties grown on the 3 farms were very similar to each other, and therefore the genetic effect on the differences in flavors across the 3 farms would have been likely minimal. So we analyzed also the temperature, pH, and microbes at the other sites. We saw that in Huila, the temperature and pH profiles were quite similar to Santander. However, in Antioquia, the profiles were a bit different. When we analyzed the microbes, we saw the fermentation time. So this fermentation time effect had the largest effect on the differences in the microbial communities. So accounted for about 50% of the variation. There was also a significant location effect. So the communities across locations were a bit different. And there was also a significant harvest effect, although the harvest effect was the least at the smallest effect size compared to the other factors. So the beans that we would have analyzed on the 3 farms, we took those beans and then converted them into liquors and then performed a sensory analysis. We saw that there were clear differences in the flavor profiles for the beans from the 3 locations. We did see, though, that the beans from Wheeler and Santander were more similar to each other compared to Antioquia. So Wheeler and Santander, the flavor profiles were mainly different types of fruity and floral notes that we detected. Whereas in Antioquia, it was mainly overripe fruit, some spice notes, but there was also some all flavors detected from the beans. Now, to better understand the link between the microbes and the flavors, we then reconstructed the genomes for the microbes from the fermenting cocoa beans. So essentially, we constructed metagenome assembled genomes, or MAGs for short. So what is a MAG? Well, when it is we collected samples from the fermenting beans at the 3 sites, in each sample that we collected, we would have had hundreds, if not thousands, of microbes in each sample. We then isolated the DNA, so the DNA would have been a mixture of these different microbes, and then we went about sequencing them. So these are like pieces of a puzzle. So we then took these sequences and then assembled them into bigger pieces. So it's taking pieces of a puzzle and assembling bigger parts of that puzzle. And these bigger parts, we call them contigs. And then we separated the contigs into groups, which we call MAGs. Right? And each MAG represents a microbe that was present in the original sample. So we were able to construct more than 1,500 genomes from the fermenting cocoa beans from Colombia. After quality filtering and removing the duplication, we were left with about 221 genomes, which range from low quality to high quality, which was pretty good. We then went about classifying these genomes, so essentially determine taxonomy. And what we saw was the microbes the genomes that we were able to assemble essentially covered the main groups of microbes that we saw in the fermenting cocoa, which was pretty good as well. We then wanted to know what was the abundance of these MAGs in the fermentations. So to do this, we then took the reads and then mapped to the MAGs, to these genomes. We saw that more than 75% of the reads, or read data mapped to the MAGs, indicating that our genomes that we assembled actually represented the majority of the data that we would have generated from the sequencing, which was really great. We then looked at the abundance of the MAGs across the different locations and different time points in the fermentations. And essentially what we saw was that the MAGs were unique and enriched in certain locations. So essentially, we saw that MAGs, we saw that the microbes, specific microbes were present in specific locations. So we then went about to understand the functions for these genomes. So to do this, for each genome, we then identified the genes it contained and then predicted the metabolic pathways to look at the functions that were enriched at the different time points during the fermentation at the different sites. Now, essentially, what we saw with this was, while we saw different microbes between the 3 sites, we saw that functions were generally conserved. So essentially, even though we had different microbes, they were performing the same functions. They were doing the same thing or similar things. However, we did identify some functions, though, that were enriched and unique to some locations versus other locations. And based on our predictions of these functions and their metabolic pathways, these functions are involved in producing different flavor precursors, likely explaining the differences we saw in the flavors between the 3 sites. So to better understand this relationship, we then went about constructing a genome scale metabolic networks for each microbe. So what exactly is this? So for each genome, we identified the genes and predicted the functions and pathways. And based on that, we could then also predict metabolites. So the metabolites each microbe can use and then excrete. And essentially, we created a network for the microbe. And we would have done this, we did this for the 200 plus microbes that we identified from the genome. So having this information now, the microbes, however, don't act independent of each other, but they are a community. So they interact with each other and communicate with each other, especially there is a lot of cross-feeding potential. So for instance, metabolites produced by one microbe could actually be fed and used as a substrate by another microbe, which could produce something else and feed back into the original microbe. So there's a lot of cross-feeding happening in the fermentation. So we then went about modeling this cross-feeding potential interaction across the community and constructed a metabolic network for fermenting cocoa. So we built our model, and so what this figure shows is the metabolic potential for the microbes. So each circle represents a unique microbe. The closer the circles are, so when they cluster closer together, their metabolic capabilities are more similar to each other. The color indicates the abundance of the microbe at the different time points. So the dark blue indicates enrichment at that particular time point. So having constructed this metabolic network for the fermenting cocoa, we saw that the metabolic potential for the community was greater than the sum of the individual microbes. So essentially, by working together, these microbes were able to reach a lot more metabolites compared to if we look at the metabolites of each individual microbe, and if you were to add that up. So there was, by cooperating, there was a huge metabolic potential. Based on this model, we were then able to reduce this community. So based on modeling, we reduced the community down to a minimal community based on identifying critical species. Because we now have this network, we identified the critical species in the network, and our model predicts that we could then take the critical species and be able to reproduce the entire metabolic range for the community. And we should be able to use this minimal community to ferment cocoa. So just to summarize the results, we saw that while there were different microbes across the different locations, functions were generally conserved. And this is likely due to redundancy across the microbes, across the community. So there was functional redundancy. Based on that, we did identify though functions that were unique to particular sites, and these functions, based on our annotations and predictions, are involved in producing flavor precursors that likely contribute to the differences in the flavor profiles that we detected across the 3 locations. We were then able to model this into a metabolic network, and we identified key species in the fermentations that are responsible for driving the fermentation. And based on our modeling, we could then take these key species and reduce the complexity of the entire network down to these minimal communities. And then we should be able to ferment cocoa. And this is something that we are currently exploring further. So with that, I would like to say that we're very grateful to the funding agencies who has been supporting this research, as well as our partners and our collaborators who've provided a lot of support. Okay, thanks for listening.
All right, thank you very much, David, for your presentation, showing that, microbial communities play such a more significant role than the composition of species in fermentation. Interesting stuff. Any questions for David? Let's see. I see a comment. Hello, everyone. Thanks for the assessment. Fermentation contributes to a lot of flavor. Not a question, but just a comment.
Oh no, we're happy for that comment. And it's very true. The fermentation does drive a lot in development of the flavors and the flavor precursors. So, and this is something that we've been exploring further. And we are seeing now a direct link between microbes and specific flavors. So, so I do think that the research is quite exciting, and it's sort of the direction we are heading in right now.
Right. Deodat Sigoveng is asking, which region produced the best flavor?
Which I'll get to my Colombian friends upset. I think so. So what we did, we did have like a survey at Nottingham, because we worked with a local chocolate producer, Louisa Vegan Chocolates, on the city center, and so she would have prepared samples, and we had like tasting sessions and did these surveys. And I mean, generally people preferred the chocolate beans from Wela and Santander more. They prefer the sort of fruity floral notes more than the spicy type notes. Although there were a number of people who prefer the Antioquia beans. So I don't know. I wouldn't say which one is better or not. I would just say that it depends on preference. So for me, what I personally like might be different from what someone else is like. And it's sort of kind of trying to tap into that, to the consumer. You know what your consumer prefers, and you could produce that. So I think that's probably the best way to go with it. So.
Sure thing, David. Just some comments as well from Ghana. They indicated that they do 6 days fermentation, and they turn beans every 48 hours and sun dry to get fine flavor.
Right.
And John Cahill is thanking you for your very interesting and important work. All right, I think that's all the time for questions for you, David. You're off the hook now, and we can now move on to the other presentation. Thank you so much.
Okay. All right, thanks. Bye.
All right, so that wraps up our area of fine flavor quality management. And now we're about to shift gears into the realm of genetic resources and conservation. Such an important topic to ensure that we can safeguard this ever so important commodity called cacao. And first up in this category, and our third presenter for today, we have Mrs. Anil Holder-John, and she's the team lead of the agronomy section at the Cocoa Research Center, who will be giving us an update on the rehabilitative work being done by the agronomy team at the International Cocoa Gene Bank, Trinidad. Mrs. Anil, the floor is yours.
Good day, ladies and gentlemen. Today, I'll be speaking about rehabilitation and maintenance of the International Cocoa Gene Bank, Trinidad, or ICGT as we call it. Before I go into detail on this work, I'll give a brief synopsis on the ICGT location, its purpose, some history, and the general operation. What and where is the ICGT, or Gene Bank as I will call it? The Gene Bank is a living collection of cocoa germplasm. It's situated in north central Trinidad and is bounded by the Caroni River to the north. This provides a source of irrigation, but also, to our dismay, flooding in the rainy season. The role of the ICGT. The primary role is conservation. Secondly, we characterize and evaluate the important germplasm held here. But in order to efficiently utilize the collected information, it is important to document and store in a way that can be easily accessed. In other words, documentation supports utilization. The evaluation of the clones has shown clones that are disease resistant or tolerant, that are tolerant to drought conditions, and some which show low cadmium absorption. These identified clones can be used as clones in their own right if they have beneficial traits such as high yield or good quality, or they can be used by breeders to produce clones that could be grown in harsher conditions or harsher environments. For example, we may have clones that could grow in low rainfall areas, or clones that could grow in slightly polluted soils polluted with cadmium. So just to summarize this slide, the role of the gene bank is to transform the global cocoa industry by being the backbone of the Cocoa Research Center, which facilitates genetic, quality, and screening research capable of making positive changes and finding solutions for challenges in the cocoa industry. The next few slides will highlight a few important milestones in the history of the gene bank. It was established in 1982, and it's approximately 37 hectares. And in 1984, the CRC, then called the CRU of Trinidad, signed an agreement with the International Board for Plant Genetic Resources to conserve cocoa germplasm at the ICGT. In 2008, the gene bank came under FAO when the government of Trinidad and Tobago, in the name of CRC, became a signatory of the International Treaty on Plant Genetic Resources for Food and Agriculture. During that time, 2006 to 2009, there was a major upgrade of the gene bank, which is funded by the Dutch Ministry of Agriculture, Nature and Food Quality, and the Cocoa Research Association of UK. This project we call the LNV project. With this project, we were able to establish subterranean irrigation systems with hydrants used to water the plants. They also constructed 2 artificial ponds that provided about half the irrigation water required in the dry season. We were able to secure the gene bank with a perimeter fence using the Pomerac windbreak, and we also reestablished some trees. The gene bank or ICGT consists of a primary collection which has over 2,000 accessions. And this genetic makeup represents more the 10 cocoa populations that exist. Besides Theobroma cacao, there are other Theobroma species conserved at the gene bank, which include Theobroma grandiflorum, Speciosum, obavatum, microcarpon. We also have some interspecific crosses and Hirania species, which is related to Theobroma. The gene bank is separated into 5 main fields, with roadways separating the fields to provide easy access. The shade trees used, initially we used a temporary shade called a Mysore banana, which is a non-commercial banana. And for permanent shade, we used the Immortel, both the Eutrinia glauca and the Eutrinia propaganda. That's it, the water Immortel and the mountain Immortels. The initial plots consisted of 16 trees, and later when we did the upgrade using the LNP project, new plots would consist of 8 to 9 trees. This was to minimize the space required and minimize the maintenance costs also. Besides the primary collection, there are enhanced populations, which enhanced populations for black pod resistance and witches boom resistance. We also have a gene-wide association study population consisting of about 500 individuals, and this is for cadmium absorption. The gene banks need support to continue its production, and some support is provided via the greenhouses that are located at the university campus in St. Augustine. We have several greenhouses: a propagation greenhouse, which has a mist irrigation system and a plant hardening facility. There are maintenance greenhouse where we keep both at-risk sections and newly acquired germplasm. We maintain these as grafts in the greenhouse, in the maintenance greenhouse. And there are 2 additional greenhouses used for screening germplasm. This includes screening for disease resistance and drought resistance. The gene bank is not stagnant. There's movement still coming in and going out of the gene bank. The transfer of germplasm is done through the International Cocoa Quarantine Center, Reading, to countries which have requested germplasm under a standard material transfer agreement. We usually have about 2 shipments per year. Between last year, July, and currently, we've had about 35 groups of clones being sent. And we also introduce clones via the International Cocoa Quarantine Center, Reading. We usually have 1 shipment coming in per year, and between July and this year, we've had about 25 groups of clones. So on to the routine maintenance and the rehabilitation work done at the gene bank. What is the routine maintenance and who funds it? The routine maintenance work is funded mainly by the Cocoa Research Association of the United Kingdom, and to a lesser extent, the government of Trinidad and Tobago. These companies, Mars, Wrigley, Mondelez, ICE, they are private companies that support the CRA and provide the funds for the CRA to assist us or to fund our routine maintenance. What does the routine maintenance entail? Weed management, which we do about 4 cycles per year. Shade management, inclusive of clearing large fallen trees. There's true-point pruning, which happens about 3 times per year, 3 cycles per year. We do minor sanitary pruning and very limited termite control. During the dry season, we have the fire trace being maintained. We have a fire watch. This year we had 2 months of fire watch. And for irrigation, we had 3 months of irrigation this year. We do fertilize a few trees, inclusive of very young trees and certain trees that are selected by researchers for use that year. This year we did about 100 trees we fertilized using routine maintenance funding. Flooding is always an issue. Sometimes, in some years more than others. For the past 2 years, we had a lot of floods. Last year we had 5 floods, and for about 11 weekdays, we had no access to the gene bank. So we spent a lot of time sometimes clearing debris after floods. This year, about 20 man-days were spent just clearing drains and around plants after floods. We have also some sheds that we do some maintenance of the sheds during routine maintenance work. But as the trees get older, they need more support. This involves more work and greater costs. With the Cocoa Conservation Initiative funding, we are doing the extra to give support that our ICGT trees require. So the Cocoa Conservation Initiative is funded by private companies. These funding companies include OFI, Cargill, Whitaker's, Guittard, and Barry Callebaut. This funding project, the CCI funding project that we've been doing for the past 2 years, includes agronomical activities that I will be speaking about in the subsequent slides. And there are other activities included in this funding. I'll just briefly mention them here now. The field evaluation for witches wound tolerance, developing a method of identifying 2 cocoa Badner viruses, DNA fingerprinting of cocoa accessions, GS mapping of the trees, and building a database. These items all assist in the management, but I will only be discussing agronomical activities. So with this funding, we did a major structural pruning was undertaken. To date, we have pruned 6,025 trees. We've also fertilized all these trees we've pruned, and the clones that are at risk, we've started propagation of clones that are at risk, meaning clones that we have less than 3 trees represented in the gene bank. And so far, by rooted cuttings, we've propagated 149 clones that are at risk, and we've transplanted 187 at-risk clones, which were propagated by grass into larger plots to be grown in the greenhouse. We've also relabeled 9,000 cocoa trees. Witches' boom disease, possibly due to climate change, has been affecting the ICGT greatly. So we did some extreme sanitary pruning and spraying, with at least 4,031 trees being sprayed twice and 1,994 trees being sprayed once. There was intense termite treatment done around the perimeter fields and also affected trees in the fields. The CCI funding was also able to upgrade the propagation facilities, including the propagation greenhouse, and start the preparation for land preparation for planting at-risk clones. So, previous slides, we spoke about what we did. This slide just indicates to us what we have to do still. And with this CCI funding, what the planned work includes comparing systemic and contact fungicides to manage witches' boom epidemic, the epidemic we have currently under, to introduce commercial biological control, and to compare their efficacy to commercial fungicides. We plan to continue the sanitation and fungicide application to all trees in the gene bank, working on improving our rooting success, continue propagating at-risk clones, replace the remaining plot labels, and replant propagated at-risk clones. Thank you very much for your attention. Any questions now?
Thank you very much, Anel, for sharing that very important aspect or rescue mission at our International Cocoa Gene Bank. And Anel, I believe we have some questions for you in the chat. Let me get that. So, question from Gabriel, how do different gene banks cooperate? For instance, do you have some coordination with Cartier ITC in Peru? How is the coordination maintained and supported?
Well, we do have programs that we associated with both of those collections, and we do also have visits. With exchange of germplasm, that actually goes through the International Quarantine Center at Reading. So before any kind of germplasm is exchanged, because we do have some duplications in some of the gene banks, but it has to go through a rigorous quarantine period. So it takes about 3 years in the quarantine center at Reading. But we do have exchange of germplasm, and we do have relationships with the other gene banks.
All right. Thanks, Anna. We have another question from Deodat. Sigoveng, have any accessions been released to the public?
Well, released, you mean like totally released? Well, we have clones at the gene bank, for instance, the ICS, which are a part of the gene bank. At the gene bank, we don't do breeding per se, the ministry does breeding, but we do germplasm enhancement work where we work to do courses to enhance the population for disease resistance, perhaps. And some of these clones we've worked with ministry to try and incorporate it into their breeding program. But to release to the public, in Trinidad, there are clones that exist in the gene bank that are also out there.
Okay, we have another question from Samuel. Does the ICGT conserve clones coming from another country? Can a country place a conservation request for its own clone in the ICGT?
Yes, we conserve clones from all over the world. We just have to make sure that it goes through quarantine and it spends time in quarantine before being grown in our gene bank. But yes, and we go through, at the moment, we use the International Cocoa Quarantine Center at Reading.
Right. Okay, I believe that's all the questions we have for you, Anel. Thank you so much for your presentation. Oh, wait, there's one more. What type of fertilizers do you usually use in the gene bank for rehabilitation? And what is their impact on the physical-chemical properties of the soil?
All right, so previously, we had minimum fertilization, but with this rehabilitation period that we're going through, we are actually selected a fertilizer that was recommended by the ministry that, they check their cadmium level. So it's an NPK fertilizer, Myla, and combined with nitro board. So at the moment, that's a fertilizer mix that we've been using at the gene bank.
Well, thank you, Anna.
On the environment is to be determined, but it has promoted some growth with the rehabilitation.
Okay, great. Thank you. You can address the rest of the questions in the chat when you do get an opportunity to do so.
Yeah, thank you, Gideon.
You're welcome.
Thank you, audience.
All right. Okay, so up next, under this genetic area, we have Mrs. Surja Chakrabarti, who is pursuing a PhD in crop science at the University of the West Indies, and she's working on drought tolerance of avoidance of selected cocoa genotypes from different genetic groups, focusing on traits like flowering time and pod development period and some of the yield components. And as part of her presentation today, she will be presenting on exploiting the cocoa genetic variation for flowering time and pod development period for climate adaptation, relationship to selected yield components. So, Surja, take it away.
Hello, everyone listening this symposium. Today, my presentation is about exploiting the cocoa genetic variation for flowering time and for development period for climate adaptation, genetic variation, and relationship to yield, selected yield components. Cocoa belongs to the family Malvaceae. It is commercially used as raw material of a billions of US dollar chocolate and confectionery industry. It is also used to produce cocoa-based beverages like tropical wines, cosmetic products, and nutraceuticals. The waste products from cocoa cultivation can be converted into animal feed and fertilizers. It is valued as an important driver to the economy in West Africa and Southeast Asia. Cocoa is originated in tropical South America as an understory tree in the Amazonian forest. Motomar et al. Identified in 2008 10 genetic groups of cocoa dispersed in tropical Central and South America, as well as 2 hybrid populations like Trinitario and Refractario. Geographical diversity allows local adaptation to different environments. International Cocoa Gene Bank of Trinidad and Tobago. ICGT. This one is situated in the foothills of the Northern Mountain Range, continuation of the coastal ranges of the Andes Mountains in Venezuela, a dry season that varies from 4 to 5 months, January to April or May, followed by a prolonged wet season, May to December. Now, climate change. Climate change is a complex phenomenon. Anticipated changes will result in changes in amount of rainfall, distribution of rainfall, and predictability of rainfall. It is expected to result in rise in temperature and rise in carbon dioxide levels. Plants have evolved several mechanisms to overcome drought. The slide shows anatomical, physiological, biochemical, and agronomical changes that can be used to generate tolerance or resilience to drought. Often these approaches improve survival, but at the expense of yield. Hence, we are proposing that flowering time and pod development period are very useful traits in avoiding drought and therefore unpredictability arising from climate change. So the objectives of this study are to determine the genetic diversity that exists within a representative subset of germplasm held at the International Cocoa Gene Bank core collection for flowering time and for development period. To correlate the selected traits for 5 consecutive years. To identify the interrelationships among the selected traits like flowering time, pod development period, on yield-related traits such as pod size, number of beans per pod, bean size, and bean mass. Materials and methods, a representative number of genotypes from 10 genetic groups and 2 hybrid populations were selected. For each accession, minimum 2 to 3 trees were selected. Flowering time, pod set, pod development period were determined by carefully tagging flowers and following them to maturity. The flowering time has been noted during the first flowering, after first rain, following the dry spell for each tree for each accession. The pods are individually tagged after pod sets, and measurements like length and width were taken in every 15 days up to maturity for each accession. Now, this is the rainfall pattern for 5 consecutive years, that is from 2016 to 2021. We can see the differences in graphical representation. The 37 accessions were common for 5 years out of 300 genotypes or accessions that were evaluated over those 5 years. Now the results. This study shows wide variation in flowering time following a dry spell among cocoa accessions selected across different genetic groups and has identified some accessions with varying flowering times. Although the environment has a role to play in the inheritance of the trait, the moderately strong correlation of accessions across 5 years indicate a genetic component. Though around 10 graphical representations were found for flowering time, only 3 are showing here. All of them showed strong correlation as well. A combined analysis of variance of 37 common accessions for the 5 years under study showed significant differences between years accessions and a year-end accession interaction for pod development period also. Here also, 10 graphical representation was found, but it's not showing here, but same moderate to strong correlation were there. For pod size, there are significant variation found for pod length and pod width among the accessions. The variation in pod length between accessions studied were greater than the variation of pod width. A strong correlation is evident among the 37 common accessions over 5 years for pod size. Here also we found 10 graphical representations, but it's showing only 3, and all of them have strong, moderate to strong correlation. Now, the performance of accessions or genotypes over 4 years compared, 4 years here because we didn't get through the bean data for the first year. That's why we are showing the 4 years for all the traits. The performance of accessions or genotypes over 4 years compared to the average over all years on the x-axis, it shows that the performance of accessions over the years was quite consistent. For the bean traits also, performance is quite consistent. Now, correlation between phenotypic traits that are studied, except for a moderate but relevant positive correlation with bean size and bean mass, flowering time did not display a significant correlation with most traits studied. Pod development period had a moderate, significant negative correlation with average pod growth rate, suggesting that the accessions with a longer pod development period tended to have slower average pod growth rate. Pod length and pod width were moderately correlated, but as expected, were strongly correlated with pod size. Consequently, average pod growth rate showed strong positive correlation with pod length, pod width, and pod size. Now this slide showing heritability. Heritability refers to how well genetic variations account for differences in traits. Estimates of heritability range from 0 to 1. A heritability near to 0 suggests that environmental variables account for almost all of the variation in a trait among individuals, with genetic differences having very little influence. Heritability is 0 for characteristics that are not under genetic control. A heritability near to 1 means that genetic differences account for almost all of the variability in a trait with very little input from environmental factors. This slide shows heritability for the trait studied. We can conclude that the significant differences with respect to flowering time and pod development period were observed. Despite large year-to-year variation in climate condition, pod development period and flowering time showed remarkable consistency across accessions over the 5 years of study, suggesting a strong genetic influence. There was evidence of genetic differences in pod development period and flowering time across genetic groups from different geographical origins, indicating that this variation could be an evolutionary adaptive response. no correlation was seen between pod development period and yield components like bean number, bean size, and bean mass, whereas very weak or no correlation was seen between flowering time and yield components, indicating that these traits can be selected independent of yield. Heritability was found in most of the traits studied, excluding pod wall thickness. Finally, the study shows tremendous opportunity to use flowering time and pod development period to select for local adaptability and drought avoidance. We are pursuing genome-wide association study to further understand the genetic basis for flowering time and pod development period. Thank you. Thanks for listening.
Thank you very much, Sergio, for your presentation, revealing how important it is for us in the cocoa industry to understand the effects of climate change. So I am looking at the chat, and I believe we have one question so far. Daniel out of Ghana is asking, what are the causes of flower abortion?
Actually, this is not my field. Anyway, thank you for the question. And actually in cocoa, that is trend, that is cocoa's characteristic, that when flower blooms, after 2 to 3 days, it's aborted or fall. So it can be the different causes, like it's
right.
cocoa's characteristics. Yeah.
Okay, thanks, Sergio. We have another question. Is it better to select a short fruit development period with big seed size or long development period with big seed size?
It depends on climate. If rainfall pattern means rainfall is very high, then longer pod development period pods can be affected from different fungus or any disease. So we can lose the pod. So in terms of high rainfall conditions, I think shorter pod development periods with big beans or large beans can be selected.
Okay, Ellen is asking, what is flowering time?
Flowering time is how we selected here. Flowering time is the first flowering throughout the gene bank, first flowering, and after the dry spell, when the first rain onset, after the dry spell, the first flowering in a tree, it's or in an accession, we found it that flowering time. Because it's this time only cocoa flowers start to flowers in particular season.
Thanks, Sergio. We have another question. Ed Seguin is asking, is there any relationship between your measured parameters and premature germination of seeds within a pod?
No, we didn't. We have to do that. Yeah, we didn't do that. For our 300 accessions, we did it. For 5 years, we didn't do that. Thank you.
Okay, we have another question from Deodat Sigubing. Have you measured the relationship between pollinator population size and climate?
No, we didn't measure that. Thank you.
Okay, all right. So thank you so much, Sergio. I believe that's all the questions in the chat.
Thank you so much for listeners. Thank you.
All right, thank you. Now, guys, everyone will have a quick video on maximizing the yield of cocoa, and then we will continue on with another presentation.
Low cocoa yields are currently a serious constraint to the sustainability of the local and regional cocoa industry. It does not have to be so. We have very good cocoa planting material of the Trinitario type available to us. Farmers just need to be able to select the best among them in terms of yield, disease resistance, and other good characteristics. Superior Trinitario cocoa planting material in the Caribbean can be found in the several cocoa producing countries such as Dominica, Grenada, Jamaica, St. Lucia, St. Vincent, and Trinidad and Tobago, which are exclusive or 100% producers of fine or flavor cocoa that attracts premium prices in the global niche marketplace. Our beans have a reputation for exceptional flavor and quality. We must safeguard that. Our Trinitario beans tend to be relatively large and plump, and violet to light purple in color when cut open. So farmers, if you see light colored beans in your field, be mindful that you could have very select planting material of a Criollo type, which produces a very good flavor. You need to monitor the production and productivity of your fields. Get to know your trees. Note which ones produce the largest pods, the most number of pods in a growing season. Cut those pods open, count the number of seeds or beans, and measure the wet weight of the seeds. So you are looking for large beans, numerous beans, and many pods per tree. Your trees must also be able to resist the prevailing diseases in the Caribbean, which are witch's broom disease and black pod disease. They should be vigorous, having large trunk girths, and they should be early bearing, so that by 3 years old, they should be producing pods. Your selection criteria for ensuring high productivity and production in cocoa are in size and weight, more than 3 grams wet individual bean weight, or 1 to 1.2 grams dried, large bean number, more than 45 beans per pod, a low pod index, which is a measure of the number of pods required to produce 1 kg of dried cocoa. Ideally, 15 to 20 pods to produce 1 kg of dried cocoa. Also look for many flowers and floral cushions on your main trunk especially. Many flowers translate into many pods. There are different plants and densities that are ideal for different commercial Trinidad selected hybrids, so that you need to know which types grow best at close spacing if you want to have a maximum number of trees per hectare, such as 1,700 trees per hectare. TSH 919 is a good candidate for close planting. Remember to replace your trees once they attain the age of 25 years, so that you will have most productive trees in your field. The trees, as they age, they will lose their productivity and you will get lower yields in the coming years. In all, once you have the right planting material with all of these traits, the modern hybrids that we have available, particularly in Trinidad and Tobago with the Trinidad selected hybrids, should give you yields of 2,000 kgs per hectare. And remember, 25% of your trees can produce up to 53% of the cocoa. You want to maintain trees that are highly productive and remove the ones that may not be as productive.
right, so let's move on to our next item in the agenda. But before we do so, please remember to place your questions in the question and answer tab. And this is because the raise hand option is not functional at this time. So please place your question in the question and answer tab. Okay, so following Sergio's presentation and under the subject area of genetic resources and conservation, we have next Mrs. Frances Bickley, and she is a research fellow at the Cocoa Research Center, a germplasm specialist responsible for morphological characterization of cacao conserved at our International Cocoa Gene Bank since 1990. Interested in all things cacao and cocoa. She will be presenting on a preliminary assessment of the putative value of leaf thickness as an indicator of drought tolerance for mass phenotyping of cacao. Mrs. Bickley, the floor is yours.
Hello, good day. I am Frances Bickley, Research Fellow at CRC. Today, we are going to present some preliminary results on an investigation to study whether leaf thickness can be measured routinely as a potential tool for screening for drought adaptation in cacao. And we all know that stress tolerance and climate resilience are particularly important for cacao. Cacao is ineffective in limiting transpiration in dry air. So a quick and efficient method with other assays for mass phenotyping can allow us to be able to select for drought tolerance in a timely manner. And this is very desirable. Physiological, biochemical, metabolomic, and anatomical or morphological changes are linked to stress memory associated mechanisms, which can be involved in adaptation to drought. And as a result, alterations in gene expression can afford us the opportunity to breed stress-tolerant genotypes using genomic tools, which are now widely available to us. The objective of this study was to identify a quick and reliable manual mass phenotyping tool for routine screening for drought tolerance at the ICGT in Trinidad. And the reason this is particularly important for us at CRC is that the conditions at the ICGT are not optimal in terms of the quality and nature of the soil, as well as the prevailing moisture conditions. The first graph shows you that for the first 4 months of the year, the availability of moisture is below what is expected or important for cacao to grow optimally. You see less than 100 millimeters of rain being available in January to April. And the second graph shows you that particularly in recent times, the average rainfall values have been less than the mean for the period 1946 to 2021. So definitely drought is a constraint. And of course, the soil type of the ICGT, it's silty clay alluvium with impeded drainage and low water retention capacity. So it is probably very significant that the trees that flourish at the ICGT have been able to do so even under less than optimal conditions. So what is drought adaptation? It's the ability to sustain biomass despite the occurrence of drought episodes during the crop cycle. So our question was, in this large and diverse collection, are there some genotypes which are able to cope with drought better than others and can still have favorable yield potential under water deficit conditions? we are not going to be looking at things like root architecture, stomatal conductance, leaf water potential, and other factors, which other researchers have been looking at in recent times. What we are looking for is a very quick and easy tool for mass phenotyping. Now, one of the considerations for the study was that plants growing in dry areas can develop what we call xeromorphic traits. And these are just traits that are structural adaptations to store water in the leaves or stems of the plant. Physiological properties such as cuticular wax can influence the plant's responses to biotic and abiotic stress and can be associated with plant adaptability under drought conditions. And some work has already been done by other researchers working on other crops, which may be applicable to cacao. For instance, Trivedi found that cuticular wax load and composition varies and can allow the plants to adapt to changing environments. Another aspect of the formulation of this research idea was the fact that previous work has also shown that the thicker the leaf mesophyll tissue, the higher is the leaf water content. And this allows the plant to be able to cope with stress to the moisture conditions. Okay, so just to refresh our memory, some of us, this is a cross-section of the leaf. It shows you the different layers, the cuticle, the epidermis, upper epidermis that is, the spongy mesophyll layer where water can be stored, the lower epidermis with the stomata, which are important for gaseous exchange. And we will get an appreciation of the fact that the thickness of the leaf can be important in allowing the plant to adapt to drought conditions. And our assumption is therefore that thick leaves may afford the plants the ability to adapt to drought stress over time and allow them to yield optimally even under water deficit conditions. And water deficit is just a state where the plant's water status is reduced. This affects the normal functioning of the plant growth and development is impaired, and of course, growth and development encompass physiological, biochemical, metabolic processes, including seed production and ultimately yield. This schematic, which was generated by Mukherjee et al. 2023, shows us various strategies for drought survival in different plants, and we see the role of cuticular thickness is recognized in this graphic. Other relevant literature included the findings of Zakaria et al. 2019, where in Indonesia, these scientists found that a higher mesophyll thickness is associated with higher relative water content in the accessions or clones that they studied. So what they found is that the leaf mesophyll thickness increases and from ICS 16 to KW 641 to Sulawesi 1, and that increase in leaf mesophyll thickness is indicative of higher relative water content. So we have this theory underpinning our study, and we embarked on a very small study because we need to replace the micrometer screw gauge that we currently have. So once we get results that seem interesting, we intend to expand the study. So for this preliminary study, we use 60 accessions, 30 with pod index values of less than 20 and 30 with pod index values of 38 or more. And for those who have forgotten, pod index is basically just the number of pods required to produce 1 kg of dry cocoa. So the fewer pods that are required, the more favorable the accession is deemed. Okay, we also looked at leaf texture to see if there was any observable, trait that could be explaining the better yield potential of some types versus others. Okay, so measurement of leaf thickness was done in a standardized manner, which is shown here, and the measurements were done between the veins. We avoided the veins, and we used the basal part of the leaf, and all of the leaves are in interflush 2 stage. Okay, so some quick results. What we see here in this histogram of the leaf thickness data that was generated is that the distribution is bimodal. We have, quite a few of the accessions with rather thin leaves of about, 0.17 millimeters compared to another group with mean leaf thickness of 0.24 millimeters. And there's a transitional group where there weren't many accessions that had leaf thickness between 0.2 and 0.22 millimeters. I think this transition is very important. So our findings through analysis of variance, what we find is that there is a significant relationship between leaf thickness and yield potential as measured by pod index. However, the positive trend is only evident for the group of accessions with unfavorable yield potential, and that is with pod index values of more than or equal to 38 pods. And shown in another way, we did a regression analysis, and here we see that all of the accessions which had mean leaf thickness of less than 0.20 millimeters, and in 2 cases, 0.22 millimeters thickness, all of these had high pod index values. So what we would call the poor performing accessions in terms of yield all had thin leaves, as opposed to the accessions with favorable yield potential of 20 or less. And we have a lack of a trend here, but most of the accessions with favorable yield potential had leaves that were thicker than 0.22 millimeters. Interestingly, a very favorable accession in terms of yield potential, ICS 68, had a fairly thin leaf. Okay, so this is interesting. Okay, and although it's preliminary, it encourages us to probe more deeply. Once we have a new digital micrometer screw gauge, we're going to do a much larger or more comprehensive study to look for robust results. Okay, so generally our conclusions were that there's a lack of a strong trend for genotypes with favorable yield potential and leaf thickness. However, there was a threshold value of 0.22 millimeters, under which we found that all of the accessions studied with unfavorable pod index had leaves that were thin and were less than 0.22 millimeters in thickness. So the results are very preliminary, but interesting, because it may allow us to select for genotypes that do not have unfavorable yield potential. It's a negative screening technique. Okay, so the take-home messages: it may be possible to do routine or mass phenotyping of leaf thickness as a selection tool for drought-adapted genotypes. Once we have a very large data set, then we would be able to say with assurance whether, what the cutoff value for the leaf thickness could be. Perhaps it will be 0.22 millimeters or less. And of course, this very quick and simple mass screening tool has to be augmented by more sophisticated methods of assessing drought tolerance, which would involve artificial intelligence. Of course, you all are probably familiar with some of these methods. Some of them are already being used, canopy biomass, thermal chlorophyll fluorescence and such. I think an important message is that we need to leverage the data and resources we have at the ICGT, this very large and diverse collection of cacao, where the input system is low, the soils are suboptimal. We could have what we categorize as water deprived conditions, where it is quite likely that we have xeromorphic adaptation evident. And we can, look at the good performing accessions and try to understand what is responsible for these accessions performing well, even under suboptimal conditions. And of course, all of this mass phenotyping must be complemented by controlled or greenhouse studies. And we can all work together in trying to realize the goal of developing climate resilient types that are high yielding under stress conditions, be it abiotic or biotic stress. Okay, so I will end here, and I wish to very, strongly acknowledge the support of our donors over the past 30 years or more, and the director and all of the people who have advised us and mentored us over the years, and of course our colleagues at CRC, and very importantly the staff at the International Cocoa Gene Bank Trinidad, in particular Timal Jagmohan. Thank you for your attention. I end here with some useful references. Thank you.
All right, thank you very much, Mrs. Bikili, for your presentation, revealing so many aspects about that leaf thickness in cacao. So we have a question in the chat for you, sorry. And we have from Samuel, I think to conduct this study, the samples were collected in the ICGT, which were all subjected with the same level of variation of climatic parameters. Wouldn't it be better if clones were subjected in a controlled environment with different levels of temperature to assess the variation of the thickness of the leaves and the variation in agronomic parameters?
Hi, good day. That's an excellent question, and it just supports what I said. Our work is preliminary. We are looking for a quick and easy routine tool to phenotype within the gene bank. But of course, whatever we do must be, corroborated with findings that are revealed via controlled studies. Controlled studies and field studies must go hand in hand before we can be assured of, the potential performance of any cacao genotype. So that is a very good question. And, I look forward for us conducting further research. In the greenhouse, hopefully if the leaf thickness is indicative of resilience in cacao, then we could routinely do screening within the greenhouse environment as well. And, then study precisely, deconstruct all of the factors that may contribute to this supposed resilience. Thank you.
Okay, we have time for one more question. How many data points on each leaf were taken?
Okay, that's a good question again. I didn't mention, but I did have a quick slide for the preliminary introduction to this seminar. We measured 20 leaves per accession at the plots for each accession, so 20 leaves. That is a conservative number, but based on previous findings, when we select in a standardized manner, leaves at interflush 2, we found that there was no very large variation in terms of the values recorded. So 20 was used in our study and would probably be used in a larger study once, well, we already got our new digital micrometer screw gauge. So we're going to start the research again and hopefully do it, on a large scale. Thank you.
And Mrs. Bikele, we have one more question we can fit in from Caleb Lewis. He's
Yes.
asking, in this study, did you quantify plant size?
No. Caleb, as you know, at the gene bank, it's not an experimental design. The trees are different ages. Some were planted in 1988, some were planted later. So, yes, that's a confounding factor. The age of the trees are not standardized. However, as I said, because we were looking at interflush 2 leaves, hopefully the age of the plant would not really have a significant influence on the observation of the leaf thickness. Thank you.
All right. Thank you so much, Mrs. Bikele. Okay, so up next, last but not least, and joining us for our final presentation of the day is Dr. Chris Turnbull. Now, he's a research fellow at the School of Agriculture Policy and Development at the University of Reading in the UK. He manages the International Cocoa Germplasm Database, or ICGD for short, which aims to collate information on the origins, characteristics, and availability of cocoa varieties, and to make this freely available to researchers and breeders. However, today, Dr. Turnbull is going to take a different slant. He's going to talk about some of his other work implementing digital data capture in the field, with his presentation aptly entitled Using Android Devices to Collect Data in the Field. Please welcome Dr. Turnbull.
Hi, my name's Chris Turnbull and I'm from the University of Reading in the UK. And I'm just going to give you a short introduction to some of the work I've been doing on digital data capture in the field. So research and breeding trials can generate large amounts of data, and detecting any errors can be difficult and time consuming, and it's often impossible to correct any of these after the event. So a data management system can improve overall efficiency by both reducing errors and improving the speed at which data is collected and becomes available for further analysis. So although I'm working on several different projects at the moment, I'm just going to concentrate on the one I've been working on the longest, which is the Mabang Mega Carrier Selection Program in Ghana. This is a large-scale cocoa breeding program that was initiated in 2005. And as of last year, there were 61 trials covering over 200 acres. So, as expected, this involves quite a lot of staff. However, the key thing is that there's only a small number that are data proficient. And so we've been using Android devices to help collect data in the field. And Android phones are widely available everywhere. And you're not tied to any particular manufacturer. In fact, you're not tied to any particular device, because although the phone's camera is perfectly good at capturing QR codes and barcodes, you can also get dedicated barcode readers, either as little Bluetooth devices that connect to your Android phone, or you can get devices with that scanner built into them. And these just allow improved speed if you've got a lot of labels to scan. And then the Kobo Collect app is free, and you don't need an internet connection while you're collecting data in the field. The forms can be remotely updated, and the data that you've collected can be automatically uploaded as soon as you've got an internet connection. Forms are also dynamic, and they allow real-time data checks. I'll come back to those 2 points later. We've also introduced barcode labels. This allows the identity of the tree to be quickly and accurately input. Although there's lots of different ways of generating barcode labels, often with dedicated printers and software, we've just stuck with the system that was already in place at MMSP, which is printing on A4 paper with laserjet printers, cutting labels out and laminating them. So we've just kept the same information as before and just added usually the location as a barcode. And then any of this data has been entered into a database with a user interface. So access is via a web browser, so it's familiar to users. And the one in MSP's on a local server, so it's on an intranet. But of course, you can host it on a server that you can access online from anywhere in the world. You can create summary reports and export files to Excel. There's various data checks that are built in, and those labels, barcode labels, can be generated in a standard format. So the easiest thing is just to run through an example. So collecting harvest data is a multi-stage process. It's collecting from individual trees or at plot level. There's pod breaking, wet weights, then the fermenting and drying of beans until eventually you collect the bean dry weights at the end. So there's different staff at different locations and at different times. And of course, there's multiple harvests per season. So one thing we introduced were these unique sample labels. And these are just based on simple numeric series. So you can see it's just 1, 2, 3, 4, and so on. And the idea is that these are printed, cut out, laminated as before. And you can just take them out in any order. And these are used to follow the pods and then the beans all the way through the harvest process. So when you're out in the field with your mobile device, the first thing it'll ask is whether you're using a mobile phone and therefore the camera to scan barcodes, or you actually have a dedicated barcode scanner. And the next step is to scan that label. So just hit that Get Barcode button. In this case, it's the camera that opens, automatically reads the QR code, although it could be the linear barcode, and that information goes straight in. The user will then just continue to collect the required information, in this case the pod numbers, and these forms are all based on the original paper forms that have been filling in since the beginning of the program. So the users are already familiar with what's required. And once you've finished collecting on that tree, you can move to the next. Of course, every now and then you might come across a tree with a label that's either missing or is damaged in some way and can't be scanned. So it gives you the option to input that information manually. And of course, because it's based on the location, the user knows the values that are required and can easily enter them, and then continue on to entering the pod numbers. Another thing you might come across in the field will be a dead tree. In this case, you select this option, the location and the time is recorded, but there's no further request to input the pod numbers. Once all the trees in the plot have been recorded, then the plot total can be entered. So all the usable pods. And so this is where we can introduce one of the data checks, where that total is compared with the sum of all the individual trees. And if it doesn't add up, then it gives you a warning. And that allows staff to see if there's any pods that have been left behind within the plot and adjust if necessary. The next step is scanning that sample label that I mentioned earlier. So in exactly the same way, camera opens and the information goes straight in. And then that label stays with the pods, pod breaking, fresh weights and dry weights. Of course, if there are no pods on the plot, then it doesn't ask you to scan in the label. And once you've finished, you just hit quit and then save and exit. No need to adjust anything. And as soon as you get back within internet connection, that data will automatically be uploaded. And similar forms are used to collect the other types of data, pod breaking, girth, and so on. And of course, that data is now immediately available to view on the screen, or to run various checks, or to export into Excel for any further analysis. And of course, all these outputs have consistent formatting. The kind of data checks can include missed trees, and this is distinct from dead trees. Duplicates, where a tree might have got more than one set of data recorded at the same time. And it can also highlight any values that are outside of expected ranges. All of these allow you just to see if there's any issues with the data at a time when you can go and correct anything if need be. Another area where the system has been used extensively is in managing the nursery. So you have a lot of plants being budded each year, and staff need to know the number that are available and where they are in the nursery. And ultimately, we need to be able to trace any trees in the field back to the original parent trees. So here we're using budwood labels, which are again based on a simple numeric series, this time with a BW prefix, just to distinguish them from other labels. And in the same way as before, these can be used in any order, and use a new label for every time you make a collection, whether that's from a different tree or the same tree at a different time, that it can be reused in the next season. A nursery label is given to a group of grafted plants when they move to the nursery, and it stays with them throughout that period, and scanned every time an inventory is taken. The barcodes generated from the numbers from the budwood label combined with 2 digits from the year. It also includes the information of the parent tree and the time at which it was collected. So the nursery has always been divided into a grid system with letters and numbers. So all we've had to do is introduce a barcode to the existing labeling system to allow it to be quickly and easily scanned. And it only needs scanning when material first moves into the nursery, and on the rare occasion it is subsequently moved. And so what you end up with is a nursery inventory. So here you've got all the numbers of plants in the different stages in the nursery process, and they're listed by clone. And of course, some clones might have been collected more than once, and so be in different nursery locations. And that little eye logo, clicking on that brings up a pop-up that tells you further information about the locations within the nursery. The nursery inventory is also updated with trees recorded dead in the field. So when the plants are ready to be planted in the field, they're given individual labels. And here they're just based on the nursery label with an additional number created from the inventory. So here there was 4 plants. And as well as having the full clone name, it also includes on the top of the label the nursery location to help staff find the correct place in the nursery. And then finally there are these labels, which are given to the plants after they've been planted in the field. And these are generated from the trial plan, and the barcodes are based on the location, so trial plot tree. And these are the long-term labels. So if they're lost or damaged, they're replaced. And these are the ones used for collecting the data in the field. You can see that this is a newer set where we've started to introduce QR codes because they're smaller and more compact, but are easy to scan by mobile devices. But some of the older devices we're using can only read the linear barcodes. They're still on there, but they contain the same information. And here you can see a plant in the field with both the labels on them. So the nursery label at the bottom that came out with the plant when it was planted, and then the top label that was added at a later time based on the trial plan. And when collecting girth data for the first couple of times, both these labels are scanned, so you can compare the expected with the actual plant in the field. There's quite a lot going on there, but of course all of those steps would have been carried out over the course of a whole year. So it's not quite as complex as it might at first sound from that quick overview. What I've described is very much targeted for the needs of MMSP. Some of the other projects are a lot less complex, with much simpler systems in place, but they all generally have 4 key components. The first of which is simplifying the identification system, so moving away from the names, which can be quite complicated, instead using locations wherever possible, or simple numeric sequences. And then that identifier is translated as a barcode on the labels. So this allows it to be quickly and accurately input, but it still allows human readable text to be included. And then of course there's capturing the data directly into mobile devices in the field. This removes the need for any further transcription, which reduces errors and saves a lot of time. Also, the information is automatically date and time stamped, and you can introduce some real-time data checks while still in the field. Then all this data is managed in a database, which is a much better storage option than Excel. It allows further data checks and summary reports to be generated in standard formats, as well as the labels with the barcodes. And of course, what I haven't mentioned before is all the names are consistent across the trials. And so I'd like to thank the Ghana Cocoa Board and CRIG and GCGRA Limited for their ongoing long-term support for this programme. I'd also like to acknowledge the previous contributions from the Government of the Netherlands, Cocoa Research UK, Mars and Mondelez. And a special thanks to Enoch Linsire, Rob Lockwood and all the staff at MMSP for their help. And so finally, I'd just like to say thank you to CRC for inviting me to give this presentation, and thank you all for listening.
Thank you very much, Dr. Turnbull. Very, very interesting and innovation at its best. I'm particularly holding up my Android in solidarity and have luck to all those iPhone users. Nonetheless, we have some questions for you in the chat. And Chris, how did the laminated labels hold up in the sun?
Hi. Yeah, so that's actually the system that had been in place going back from the beginning. So they were using exactly the same laminated labels without barcodes to label plots rather than individual trees. And so they'd been on there for certainly many years. So they do eventually need replacing. But I think we had a conversation when I was visiting that actually half the time it's actually what you're tying things to, what you're using to tie labels onto trees are often the cause the issues rather than the labels themselves. Plus, I think actually the labels are often in the shade. So under the smaller trees, there's sort of a reasonable amount of leaf. So it's actually sometimes difficult to find the label. And once the tree's bigger, it's sort of under its own shade. So actually, I don't think that they tend to miss direct sunlight. But they do last years, yeah.
All right, thanks for that. We have another question from Jeff. Samuel, he says, very interesting presentation, and hope that you can share your email with him in the chat.
Yeah, so I noticed that one, so I'll put the link. So actually that QR code that came up and the link on the talk gives you information about some of the other projects at Reading, including this one. And on that website, it has a link to my email. So I'll share that again afterwards on the chat.
All right, great. So we have a question from Romina. And the question is, if I can find it, give me a second. How long do the labels last in the field, and do they become mossy?
I haven't seen them mossy, and it's actually something I'm trying to keep an eye on, because some of the other projects are using different methodologies for generating labels. So some are using almost like credit card, effectively, that size plastic that's getting labels printed on. In the National Fruit Collection in the UK, we're doing a similar thing, using sort of plastic labels, again, that are adhesive, all of which we're assured works long term, but it's something we're going to track. But certainly those, so far, the one I know works well are those laminated A4 laserjet labels they've been using in the MMSP project for many years.
Mm-hmm. So we have been having issues with the attachment of labels on trees, and it's always a big issue. So they can be lost with time and conditions, as you will already know. What was used at the MMSP and what is recommended?
I don't know if I'd be qualified to recommend anything at this point, and I'd have to double check what they were using to attach to the trees at MMSP, because like I said, I sort of came into the project and basically adopted their current system, so I didn't need to pay too much attention to what they were doing, because I'd already figured that bit out. I was just effectively adding a barcode to their existing label. It'd be something I can easily find out, but yeah, I don't know off the top of my head.
No thing. Can this be used by farmers to collect data on farm as part of a participatory research program?
Yes, it would need a little bit of organizing with somebody. So we have another project in Ghana with sort of the looking at soils, and there it's the extension services. So although it's on farmers' fields, it's I think it's Craig staff are coming in and doing sort of the recording, but you can set it up with a barcode. So once you've developed something, you can set it up and you just share a QR code, that once they've downloaded the app, they scan that and that sets it all up. So you could easily actually put it out there, if you had sort of a central service that sort of collated the data, because that's kind of in some ways the biggest step, designing the form to do something useful and then pulling the data and analyzing that. But yes, letting people go and collect the data themselves on a form would actually be quite simple to do.
Right. And I'm not sure if you completed answering Jeff Samuel's question, but he asked as well, do you have any document or article that gives the exact methodology for the label identification in nursery and in the field?
Well, I suppose that the short answer is that it's very much location specific. So how we're tracking the plants through that nursery is slightly different to what we're doing in a different nursery, just because the way the nurseries work as such. I'm trying to think if there's anything. So there's information, like I say, on the website, and you can contact me through that as well. So that's probably the easiest thing.
Okay, great. Just to remind participants that we are not facilitating raise hands in terms of audio questions or vocal questions. You can also type your questions in the question and answer tab or in the chat, and we can facilitate responses from there.
Yeah, so I will go back and have a look at it if I've missed any as well.
Okay, great. Are there any other questions? So we'll probably give a few seconds again, just in case participants want to type their questions in the chat. Okay, well, you can always respond to them if they do appear, Dr. Turnbull.
Yeah.
All right, thanks you
Then.
so much for your presentation. Okay, at this point, this more or less concludes all of our presentations for today. And I would like to invite Professor Umaharan to wrap things up and give us our closing remarks. Professor Umaharan, are you there?
Yep, I'm here. Right. Thank you, Gideon, for a fantastic job that you did today chairing the session. And thank you all for the speakers in today's session. You all did a very good job. And I think the talks today were started with looking at the provenance of fine flavor cocoa that Lambert Motilal spoke about, and indicating that there may be a genetic basis for quality of cocoa based on his work. And then David Gopalchan came and provided some other insights as to even when the varieties are very similar across locations, you can get very different flavor profiles, as he had found in Colombia. And he indicated that that is a function of the microbial populations that are peculiar to those regions. And he has actually created consortia of those microbial populations collected from the 3 locations to validate by using those microbial populations, he can get back the same flavors he got from these different locations. And that would be a very exciting study that shows that in addition to the genetics, there may be microbial influences that contribute to quality. So I would like to open the floor for any other questions with regard to the flavor. I know that persons have also shown the effect of the location, the growing environment, also contributing to the flavor profile. So flavor is a function of different things. Genetics is important, but it's also the microbial population, the fermentation and drying methodologies used, and the growing environment that contributes to the flavor profile. So if there are any questions, comments on this topic, it's a very exciting topic. We are trying to unravel the mystery of fine or flavor cocoa at the Cocoa Research Center. So it is almost like 3 blind men touching the elephant and describing it in 3 different ways, right? One person saying that it's like a big lump of or big tree trunk because he touched the legs of the elephant. Another one looking at the trunks saying that it looks like a wiry, something very wiry and flexible. and somebody looking at touching the tail and saying it's very hairy, right? So I think different people are looking at it different angles and trying to unravel the mystery of fine or flavor cocoa. And being a mystery itself is really good, because it's a science and art of producing this fine flavor cocoa. So I think, is there any questions? Let me see in the chat. So far, I'm not seeing any questions. Let me look at our WhatsApp group to see. Nobody wants to comment on the mystery of flavor. That's very good. Okay, so it will remain as a mystery for a while until we further do further evaluations, right? So I have a question unrelated to this from Deodat C. Gobin, who is asking a very broad-based question, right? Saying, describe your ideal cocoa tree, right? So ideal cocoa tree, well, I think, I don't know the question is for me or for any of the persons who actually contributed to these talks today. But let me take a first stab at that. The ideal cocoa, there's no ideal cocoa tree, I would say, because what is ideal for one location may not be ideal for another. And so therefore, selecting a cocoa tree for an environment, for a production methodology that a farmer uses, a farmer management systems, it's all very, very nuanced, you know. So a tree that does really well in an experimental plot does not always do very well in farmer environment or farmer management. So that is why we do conduct different kinds of trials. We do trials at the experimental stage, then we take it to different locations and do multi-locational trials. We do the trials over different seasons to see how they respond to different seasons and conditions. And then we end up with farmer field trials to see how these varieties perform under farmer field conditions. So, that question is very complex. And in one location, they may desire a particular flavor profile. In another location, they may desire another flavor profile, right? So I think it's as complex as it could be. That's my best answer I can give to Deodat C. Gobin. So, so that I'm getting some comments saying this conference is very important as a farmer, and I learned a lot. Thank you, Daniel. Excellent work and well informative. Thank you, CRC. From Geeta Peters. So I think there's some good comments coming from, and there's a question from Alima Ali, you know, is it possible for cocoa to be organic? Well, at present, most of the cocoa that is produced in Trinidad is organic because of the fact that very little chemical sprays, fertilizers are used in cocoa production. But always, unless you really manage your organic production systems in a very effective way, your yields can be compromised. So there's also a negative side to it. But that's a very interesting question, and it opens up further questions. So that is, so then we went on to look at the gene bank. And we also identified potential sources of adaptation to conditions in the gene bank. That was by Surja. We also looked at the management of the gene bank, the several risks that the gene bank faces, and the management practices that are being done to manage these risks or minimize these risks by Anil Holder. So, and then we looked at exploiting the gene bank for drought tolerance, some of the traits that may be important for drought tolerance by Francis Beckley. And that's another way of exploiting the gene bank for drought tolerance. Then we also looked at labeling and managing how to better manage the information, fidelity of the collections, and it was a very good illustration of how a breeding program is being managed through barcoding system by Chris Turnbull. So I think we had some excellent presentations about gene bank management and utilization on the second half. And is there any questions, comments people want to say about management of gene bank from their experiences from national collections management? I know there was a comment about a number of the national collections not being particularly managed pretty well, and there was a request of how can materials be sent to the international collection for management. And I think there were a lot of comments about it, and from the presenter indicating that we are welcoming any such transfers to the central collection here at the International Cocoa Gene Bank in Trinidad, but it has to come through a quarantine facility to prevent risks of new diseases being introduced. So that is in a nutshell that this is today's presentations was about. Thank you all for being part of this presentation. And I will also like to thank all the sponsors and supporters for this presentation. All right, so I think I'm going to just do a last check on my network here. So there was a question that coming from Lyndon Campbell. What is the most common observations across the Caribbean with regards to cocoa production and recommendations for best practices? So this is a very good question with regard to improving best practices. So I think we have worked across some countries in the Caribbean, so I cannot say for all Caribbean countries, but generally most of the cocoa production in the Caribbean is based on small farms, mixed farms, not only producing cocoa, but other crops as well. And always the productivity levels of cocoa has been low. We usually calculate the productivity based on per tree productivity, averaging it across trees. And generally the levels are quite low. And that is because most production systems have, the trees are sometimes very old. The varieties have not been changed, although new varieties have been introduced. So we looked at the planting material pipeline, for instance, in Dominica and Jamaica and Trinidad, to see how we can improve the planting material pipeline to introduce better varieties, newer varieties to farmers so that production can be increased. We also looked at rehabilitation of existing cocoa trees from the farms to improve productivity. So productivity is one challenge that the region faces. The risks is another. Pest and disease risks, it varies depending on the countries. In St. Vincent, St. Lucia, Trinidad, it's witch's broom and black pod. If you go to the north, in Belize and Jamaica, it's frosty pod and black pod is the problem. And so I think it's been a mixed bag. The productivity is low. Managing risks is very, very important. A lot of the work that we have been doing at the Cocoa Research Center is to look at ways of managing these risks. And some of the presentations today would have addressed some of the practices, some of the things that we are doing. We had worked on risk mitigation with cadmium in the soil. We worked on risk mitigation with diseases that you would have heard. But transferring the practices, one way is to do these symposia, which we are doing, but some of the information has to be also taken across by extension to the farmers and introduced into the farm practice so that actual changes can actually happen at the ground level. So So it's a good question again, but it's a complex one. So I think there's a comment from Daniel Paroma from Bolivia, who has been collaborating with us over the years. He says that these 2 days have been important for the cocoa research team in Bolivia to continue advancing the search for genetic materials with productive potential, quality, and tolerance to other factors. We thank all the speakers for the presentations. Thanks, Daniel. Elizabeth Montano from Trinidad, she says, thank you very much. All the presentations were fantastic. I want to know what are the suggestions as we look forward to the next crop improvement of yield. Gabrielle Metz, I think, I hope I am pronouncing it well. Thank you. Hope to have the opportunity to visit on site once at least. Very interesting. Karen Lelam says, another insightful day. Congratulations. Thank you for CRC team and Dr. Ram Tahal, who did an excellent job. So, so if there are no more suggestions, questions, I'm checking my thing, seeing if there are any. All right, so I think I'm not seeing any more questions or comments. So thank you all for attending today's session, and I'd like to thank again our sponsors, Government of the Republic of Trinidad and Tobago, who have supported us over the years, the Cocoa Research Association of the UK that provides support to the International Cocoa Gene Bank and for some of the work that we do, and also the other sponsors who support, industry sponsors who support research at the Cocoa Research Center. Thank you, and we'll continue to provide updates on a yearly basis. And thank you all for being here today. Bye for now.